So this is Josh Quick's protocol, and based on our understanding, the Nanopore can do long reads, but this one it has been designed for around 400 BP PCR amplicons. So it's like amplicon sequencing with a whole genome coverage for SARS-CoV-2. So uh, how do we go about this? Like perhaps I mean, there's no point if I just read this. Perhaps you can go directly to your inputs, Ralph.
Yeah. So I'm not super familiar with the version 3 protocol. I've been using the version 2, but I know all of the major steps that goes
Yeah.
into it. And so I can tell you a little bit about some of the modifications that I've made that apply to this. Just a quick question. For the samples that you're planning on sequencing, do you guys do the diagnostic sequencing as well? Like, do you
Um,
have CT values for those?
yeah, actually, these are clinical samples, so it has been validated as positive for SARS-CoV-2. But we can also do the test again in our lab to see if there are changes with the CT values, because we expect not all of our collaborators have proper storage. For
Mhm.
instance, I'm not sure if every lab had a negative 80, so there might be issues with RNA degradation. So most probably we will recheck the CT values in our own lab.
Okay. That's good to know because we've had that same issue in that we get all of our samples from a clinical diagnostic lab. However, we revalidate all of the CT values because when we do the transporting, they should be stored on dry ice.
Yeah.
However, that's not always the case. And so some of the samples do degrade during transport. And what I found was in order for this protocol to work efficiently, your CT values should be at a value of 30 or below.
30 or
Sometimes,
below.
yeah. So sometimes you can get lucky if it's a CT of 30 to 35. However, what we found is that when the CT values are too high, you only get partial genome amplification. And so you get maybe from 95% coverage and above, and it drops down to 60 or below when you get a CT higher than 30. And so for your first couple of runs, I would say if you wanted to maximize the number of usable genomes for assembly and publications, you should stick with samples that have a CT of 30 or less. And the lower the CT value, the better. In fact, I've found that a CT of, let's say, 27 to 30 will usually give 90% coverage or more. And then anything below 27 usually gives good enough coverage for publication, for uploading to a database. But for any sample that is 30 or below, I run it through the first two steps, it looks like for this, where you are essentially converting it from RNA to cDNA. And so it looks like that's what this first step is. And it's just for any sample that's 30 and below. I just run it through this. It's not a guarantee that it's going to yield enough cDNA to sequence, but it's a good first step. And there are other checks that you go through as well to make sure that what you're sequencing is going to be high enough quality that you'll get good results. And so let's see, yeah, for step 2, I would say just follow it as is. For the CT dilutions, I've never had any samples that were higher or that were lower than a CT of 18, and so I just follow it as is. If you end up doing a dilution, I don't think it's going to hurt it at all. But one of the things that I do notice is that further on in the protocol, there's a check, and sometimes a check uh, will yield multiple different bands instead of the one band that you're expecting. And so if that happens, then it might just be that your sample is too concentrated. Uh, but more likely than not, if you end up not doing any dilutions, if your CT values aren't low enough, it's still going to work just fine. And then for that, I run it as is. For step 4, are you guys getting your primers for the amplification like as individual sets, or did you order the IDT
We ordered the IDT.
Okay, perfect. Yeah, I've not had an issue with IDT at all. And so usually what I do is I make a lot. I do the one, it comes in 100 micromolar already. And then I just make a lot of 10 micromolar concentrations for use. Uh, and then I just store it at -20. And then I just defrost as needed.
Did you have issues with perhaps primer degradation through time?
Uh, for the IDT, I haven't noticed any major degradation. So the primers that we've been using, it's about 6 or 7 or 8 months old. And so it's usually not the age of the primers that really determines whether or not it's degraded. It's the amount of times that it's been defrosted. With the number of samples that I'm working with, usually it goes through no more than 2 freeze-thaw cycles. And so the degradation, there is probably some degradation of the primers. However, it hasn't been significant enough to not amplify anything. But I would say for our general rule of thumb in lab, we usually don't go through that many freeze-thaw cycles. And so usually I store it, depending on how many samples you plan on running per amplification, doing like 50 or 100 micromolar or 100 microliter aliquots at 10 micromolar.
We usually set a limit of not more than 3 freeze thaw cycles, but perhaps we can reduce that to 2.
Yeah.
Um,
Yeah. So this is the dilution, and yeah, I just do that in PCR clean water.
So this is the Mutapex.
Yeah. So this
I think we're using the M0494.
Okay, yeah, this is the exact same one that I use, the M0494, and I follow it the same way that it says here.
So 35 cycles is sufficient.
Yes.
Yeah. Have you tried using a lower denaturation temperature?
I haven't tried a lower denaturing temperature. I know it's a little bit higher than what other polymerases use, because traditionally it's like 95. However, I did look it up, and it is actually supposed to be run at a higher denaturing and annealing. And that's just because the polymerase that they use, it's a Q5 polymerase. It's not a regular Taq polymerase. And so it needs a higher temperature in order to work properly. I haven't tried it at a lower temperature. It still might work. However, I've just used it as written and have gotten really good results.
So here, PCR dilution.
So for this is where it really depends on how high of a quality of the sample that you have. And so for this, it says to like assume that it's around 100 mass percent. That is almost never the case whenever I do my samples. It might just be because the samples have degraded. Do you guys have a qubit in your lab?
Yeah.
Okay, perfect. Because I usually do qubit. So what I do is I take all of my samples, I just pool the reactions from pool 1 and pool 2 together. And then what I would highly recommend is first off, just doing a gel, just to confirm that the samples do have bands at around 400 nucleotides, which confirms that you had amplification for SARS coronavirus 2, because 400 nucleotides is the approximate size range that you're looking for all of your amplicons. And if you see a band in there, then usually the concentration of those amplicons are high enough to give you good values for qubit, as well as good reads for the sequencer. If you don't see a band, then I just don't even continue with that sample and I put it aside. And so I do the gel first, just to confirm that everything that amplifies is good, and then whatever doesn't amplify, I leave out. And then after that, it's important to always run a positive and negative control for these assays for this specific reason. And so the positive control that I use is from Twist Biosciences. They're a biotech lab here in the US. They also have ATCC But whichever one that you go with, if your positive control is like the full length SARS coronavirus 2 RNA genome, usually I run that. And then when I do my qubit, what I do is I measure all of my samples, I measure my positive control, and then I measure my negative control. The negative control is important because there is usually excess primers and maybe some primer dimer in there, and that it will still give you a reading. It's usually around 20 nanograms per microliter on the qubit. And so if there's a sample that's like, say, 50 nanograms per microliter, and the other samples are in the hundreds, then I know for that 50 nanogram per microliter sample, there's not much amplification that actually went on. In fact, a lot of it is just background because it's close to the value that I get for my negative control. And usually it's very clear as to whether or not a sample amplifies or not, because for samples that amplify very well, they'll have a qubit value in the hundreds of nanograms per microliter. And then usually if it gets that high of a value, it will give you a very good sequence results. Anything lower than that is really dependent on how much DNA you get. And usually my cutoff is around 25 nanograms per microliter. And so I have something less than that, then I don't go forward with it. And then if it's above 25 nanograms per microliter on qubit, forward.
We actually don't have a positive control for this, but we can run a negative control. Are there any potential problems that we might encounter since we don't have a positive control for this assay?
You shouldn't, as long as you do the gel to confirm that you have amplification of your target sequence, then I don't see that not having a positive control would be any issue.
How about the upper limit for the initial concentration? Because when we did for MySeq before using the Illumina platform, there's kind of a range that we shouldn't go.
For Nanopore, the range depends on what type of DNA you're using. Usually it's 200 femtomoles. And so usually I would have to calculate femtomolarity for all of these samples because it's not necessarily how much DNA concentration you have, but rather how many strands of DNA you have in general. And so I've done this a lot of times. And so you could just base it off of concentration. And so when they say you dilute down to 10 mass percent, that's equivalent to 10 nanograms per microliter. And so based off of the qubit values, I dilute it in nucleus-free water down to 10 nanograms per micro, and that's around 200 femtomoles, which is the upper limit for nanopore sequencing. I've actually started to increase that amount because for some of the protocols, they can actually go up to 300 femtomoles and not have any issues. The reason you can't go higher than 300 femtomoles is because your limiting factor, at least from what I've been doing in the past couple of weeks, is that your limiting factor is the reagents that you put into the reactions for the library preparation. And so if you have more than 300 femtomoles, what happens is that there's not enough barcode or there's not enough adapter to properly ligate onto your samples. And so you get a lot of reads that are unusable because they don't have the complete barcode and adapter ligation. And so anywhere between 10 and 14 nanograms per microliter will be at the upper limit, but not too high that you'll get weird results.
Cool. Here, for native barcoding.
So this, they actually decreased the volume from, let's see. I'm just checking my notes here. So they decreased the amount of reaction that you put in from 5 to 3.3 microliters. You might be able to do that, but what I would recommend is increasing the amount of PCR dilution from the previous step from 3.3 to 5 microliters.
3.3. So from this is to increase template, right?
Yes.
Okay. Um, Anything else?
And then to keep it an even 10 microliters, just remove like, what is it, 1.7 microliters
Yeah,
from
from
the amount of
water.
nucleus-free water. So to keep it at a consistent 10 microliters.
Mhm. Okay. So, Khalil, we have 24 barcodes, right?
Mhm.
Yes.
Am I correct?
We have
How about when you're using flow cell? Sorry, Ralph.
Mhm.
Like, did you have any issues with contamination from prior runs, like when we use the flow cell after washing?
So for Nanopore, what they say is that the flow cell wash kit, it removes the majority of the library from the previous run to reuse the flow cell. Uh, from what I've seen, there's still some carryover of your previous library into the next sequencing run. And so when you do your wash for your flow cell to reuse, what we've seen is from some of the figures that they had, and this was like a year and a half ago, there's still about 10% retention of your previous library that may still exist in the flow cell even after washing. And even though they have nucleases to degrade the majority of the library that was left in there, uh, there's still going to be some carryover. And so, as a basic rule of thumb for that, what I suggest is if you are planning on sequencing something else, like for example, if in the near future you plan on sequencing some African swine fever, it's best if you reuse the flow cell if your second library doesn't have any samples that were, say, the same organism from your previous run. And so, for example, for your first run, you might do all SARS coronavirus 2. And then for your second run, it might just be African swine fever virus. And the reason for that is because if you have some SARS coronavirus 2 data that leaks into your second run because there's still residual library within your flow cell, then you know that it's not necessarily due to your library preparation, but rather it was just some carryover from the previous library, and you can disregard it because you know there was no SARS coronavirus 2 in that sample. There might be a way. The alternative as well is, like, for example, for your first flow cell run, if you only do barcodes 1 through 12, then in your next run, if you sequence another 12 samples, just go ahead and use barcodes 13 through 24, because that will differentiate it a little bit more. And so you can still separate what was from the previous run and what was from the new run. But if you were to reuse a flow cell, like for example, if you did 24 samples on the first sequencer, and then on the second sequencing run, you did another 24, but they're all SARS coronavirus 2. I don't know exactly how much carryover would result in, like, say, SARS coronavirus 2 from a different specimen leaking into your new sequencing run because of that carryover, because they would essentially have that same barcode.
Um, can I ask Matthew, if how would this affect? Uh, I mean, I understand if it's just 10% retention, does this affect your assembly, your consensus perhaps, or your the pipeline in terms of how they identify potential variants?
I think Matthew just stepped away from the keyboard. He said he'll be right back in the chat.
Okay. I'll just ask this again later. Because if I remember correctly, I think it was Dr. Amanda War who mentioned that if we wash the flow cells longer, like I think that was 30 minutes, the carryover would reach to the point that it's negligible. But I'm not sure in terms of how it fares. For instance, because identification of variants are very uh, tricky, since the pipeline just produces one consensus sequence per sample. So carry over. Paul, have you done readings on this?
Well, honestly, not yet. I think it has more to do with the sensitivity of the sequencing so that you can capture the variants. But aside from that,
Yeah. Also, right now we don't have enough reagents to reuse the.
Yeah, the
so
flow cell.
doesn't matter right now. Cool. By the way, Matthew is back.
Okay. Hey Matthew, I have a question. We were discussing earlier the consequences of reusing flow cells. For instance, if you use the same barcodes, and according to Ralph, there's around 10% retention of the libraries. My question is like, does this affect the bioinformatics pipeline in terms of um, uh, the sensitivity of, let's say, variant identifications?
Only if, as Ralph mentioned, you have, let's say that you run SARS-CoV-2 using barcodes 1 through 12, and then you do a flush and you run another run, also you're doing SARS-CoV-2. There's no way that I could guarantee that every single read that you're getting is from your new library prep. There's that possibility, which is why, as Ralph said, we'll always do very disparate organisms, SARS-CoV-2 or influenza, and then
Okay,
like a
but
double-stranded DNA
so
virus.
the 10% is still a big factor, so it can still affect the consensus. Is that what you're saying?
Only if you use the same, try to sequence the same organism twice. And there is a new protocol for nuclease flushes on the Nanopore site. I don't know if Ralph has employed that one yet. I know we were talking about it at some point last year, but we don't have the new kits.
Okay,
but yeah, essentially they flush the flow cell with nucleases, so anything that remains in there hopefully is enzymatically cleaved into small pieces and no longer sequenced.
Cool. Where are we again? So, Ralph, do you have any for the washing, like this proportion?
Yes. So for step 11.6, I would double the amount of beads that you put. So instead of doing a 0.4x volume, do a 0.8.
0.8.
Yeah. The reason being is I find that there's still, because they skipped the cleanup step prior to the library preparation, there's a lot of junk in there that you want to try and get rid of a little bit better. And so what I found is if I use this particular volume, what ends up happening is there's going to be a lot of excess barcode that is just carried over that doesn't have an adapter or that doesn't have a sequence from the sample associated with it. And so what you get on the sequencer is that you get a lot of adapter reads, and it may have some barcode, but it doesn't actually generate any reads for generating the consensus. And so, oh no, sorry, disregard that. That was the next step. For this step, the reason why you do 0.8 is because if you do a 0.4x, you lose a lot of your DNA. So it's simpler than that. And so by doing a 0.8x rinse, what you end up doing is you rinse out the same amount of junk, like the primers that were in there, some of the excess barcode, but you retain more of your library. And so you just get a better yield, and as a result, you get more reads per sample when you do the sequencing.
Cool, that's nice. Because they just put it here. It's really important to really optimize the process. Unfortunately, we don't have extra reagents to optimize the process on our lab.
Yeah. And then what I would suggest too is for this SFB, the one thing to note is that they don't give you extra SFB in the sequencing kits. They only have 2 SFB tubes in there. And so that limits the amount of runs that you can do per kit. What you can do is there's 2 things that you could do. One is they sell SFB separately. I think it's like $50 US for, I think 8 to 10 tubes. We haven't actually ordered it ourselves because We have a lot of leftover kits that are old that we just take the SFB from. But do keep this in mind if you only have, say, the one LSK 109 kit available. You might run out of SFB sooner than later, because usually this is only used once in a protocol, but because they're doing it twice, you're essentially using twice as much volume for this SFB. If you do want to try and conserve some of the SFB, what you can do is just repeat the ethanol rinse twice to replace one of the SFB rinses for 11.9. And then for that ethanol rinse, I would recommend increasing the concentration of ethanol from 70% to 80%. And this is something that I learned from a colleague, Dr. Annalise Ducluseau. She's been doing a lot of nanopore sequencing. She is a collaborator of ours that lives up in Fairbanks, and she's the one who taught me a lot of this stuff. And by increasing the concentration to 80%, you get a cleaner library, and you'll find that you have less ethanol carryover because a higher percentage of ethanol tends to evaporate faster from the beads. And so you get less of that ethanol retention.
During the QA steps, do you have any thresholds that you can suggest, which as identifier if we did a good run or a pretty bad run?
For step 12, I actually skipped that step. I find that it's not really necessary. As long as you, I would say if you wanted to make sure, because I know you guys are restricted by the amount of reagents that you have, I would say if you just wanted to run the assay, just to ensure that you have some yield of DNA in there, I would say as long as it's not super, super low in terms of concentration, it should be fine. Because what I found is that the fluorometer measurements, when it comes to the actual library, aren't super reliable. And that I've actually had some library preparations where at the end, I only had maybe like 1 nanogram per microliter for my concentration. But that actually gave me one of the best results that I've ever seen for my library preparations. And so it just really depends on the quality of your samples at the time. So I would say as long as you have some samples that have like at least a couple nanograms per microliter of DNA in there, then it's safe to proceed. But usually what I found is that this step 12, isn't super important because I'm still able to run through the entire protocol and still get results.
Interestingly, we had the same experience when we did Illumina MySeq. We had some samples who had really low concentration measurements from qubit, but it turns out they had a much better quality of the assembly compared to samples who had a much larger concentration prior to sequencing. So we're kind of wondering why this is the case as well for Nanopore.
Yeah. For me, what I found for Nanopore sequencing is I believe the values are artificially inflated based off of the amount of barcode and adapter that is in there. Because if you don't properly wash those away during the steps, what happens is you still get some retention from those that kind of act as filler for your library. And so you might have something that has a very high concentration, but you'll see when you actually load it into the sequencer and you go into the software, you'll see that you'll get a lot of reads that are adapter. And so usually what I found is that there's an inverse relationship between concentration and the amount of adapter or like junk reads that I get. And that the lower the concentration, the less or the higher the quality of the sample because there's less adapter or barcode that's just filling in that space. So I'm not super familiar with Illumina, so I don't know if that's something that's similar for your adapters and stuff like that, but that's something that I've noticed for our Nanopore stuff.
Where are we again? This is just part coding. So here in this is ligation.
Yeah, this step I follow the same. So no optimizations there. For step, for step, I think 13 points. Could you scroll up a little bit more? Yeah, 13.3. I don't do a one-to-one because what this results in is you have a lot of adapter retention. And so I actually do a 0.4 concentration in this step. And so instead of adding 50 microliters, I do 20 microliters. And you might have, like, you might see that your concentrations for your libraries are lower, but that's because you have a lot of the adapter step. And I found that if I stuck to this one-to-one ratio, it doesn't get rid of a lot of the adapter that's still in there. And so you have like maybe 30 to 60% of all of your reads just being adapter or like the unclassified reads.
Sorry, it's 0.4, so that's 20 UL.
Yes. Yeah, and that's actually a modification that I took from the original Nanopore protocol. And so I don't know if you guys have looked at the Nanopore community site where they have all of the protocols uploaded. They actually made changes to this to kind of improve the sequencing success rate. But I found that some of their optimizations aren't necessarily the best. And so that 0.4x ratio that I suggest is actually from the original Nanopore protocol. And it seems to do a much better job for actually getting usable data. And then for this, for the previous Ampere bead cleanup, you could substitute with ethanol. For this step, you don't want to use any ethanol. So this is the part where you do have to use SFB.
In terms of sample handling, handling the libraries, the vortexing, do we have to, be cautious when do we pull vortexing of these libraries?
As a general rule of thumb, I do not vortex because for this particular instance, because your amplicons are so small, vortexing should not affect them in any way. And so even if you do accidentally vortex, it shouldn't harm your library. However, as a good rule of thumb, like for example, if you were to work with genomic DNA, you do not want to vortex because that will for sure fragment your DNA for loading it into the sequencer. And so what I usually do, let me get a tube, is whenever I mix my tubes, I will just flick it with my finger like this. And then that should be sufficient to resuspend everything and get it homogenized. And then if you do decide to spin it down, just use a small benchtop microcentrifuge. Just spin it down for 1 to 2 seconds, just enough so that all of your liquid pools down to the bottom. And then that should be good. But you always want to avoid over pipetting your samples and vortexing, just as a general rule of thumb. That way, if you do have samples that are like prone to fragmentation from those steps, then you've already trained yourself to not do that in general. And so it shouldn't affect any of your libraries.
Cool. Yeah, so here actually, this is the part where we're kind of it's really new for us because we haven't used MinION before.
Okay, let me get Have you guys seen a flow cell before? Like, are you familiar with like how the flow cell is loaded and stuff like that?
We've seen it in videos.
Okay. Did you see the instructional video where it shows you how to load a flow cell?
Yeah.
Okay. So the video is actually pretty close to what you see in the actual experience. And so when you're handling the flow cell, it comes in like this. And so you'll see that there's like some foam on the bottom, and then you have your ASIC chip here as well. You just want to make sure that you don't want to damage any of those pins or contact points for your Nanopore flow cell. And you can pick it up and you can handle the back just fine. And so This is a used flow cell, so it's not going to be used for any sequencing. But whenever you handle it, there's some liquid that's in here and it's not going to splash or anything. But whenever you store your flow cells and whenever you take it out, you don't want to do it like upside down and you don't want to store it sideways. You always want to store it flat so that all of the liquid just stays in there and nothing spills out. But it's pretty durable. Like we've taken it to Ukraine where Eric and I have just taken these, we've thrown it into our backpacks and transported it, and it's fine. One thing I suggest is when you do plan on sequencing that day, 30 minutes or so, or a little bit farther, at least 30 minutes, to take the flow cell out of the fridge and just store it at room temperature. And this allows it to come to that 37-degree mark a little bit faster, and you'll get a better sequencing run because it doesn't take as long to calibrate. And so I would say if you're planning on sequencing, when you start your last library cleanup, which is going to be after your adapter ligation, so this step that we're looking at right here, that's when I usually take out my flow cell, because by the time I'm ready to load my library, it's warm enough so that I can actually do my QC. And then before I do any of the flush buffer and flush tether, I just want to make sure that I have enough pores for sequencing before I even waste reagents. Because I've had situations where I've prepped all of my reagents and I've combined everything, but then I didn't have any flow cells that I could use for sequencing. And so I would essentially have to wait because I can't sequence. And so once it's been like 30 minutes and you've allowed it to come to room temperature, your first step should always be to QC your flow cell. And so like on the Nanopore site, they mentioned how to do it. On the Minos application, it goes step by step on how to do it. And so I would always QC my flow cell prior to continuing forward, like doing the flush buffer, flush tether. And then what I always aim for is the more pores you have, the better. I think the maximum number of pores is 2048 or somewhere around that value. These flow cells actually store pretty well nowadays. And so even if it's a flow cell that's 6, 7, 8 months old, I usually still get 1,800 active pores or more. And you'll get that value after you do your QC. Sometimes if it's had some issues with transport or storage, you might have a lower value. And so for SARS coronavirus 2 sequencing, because these are precious clinical samples that are hard to get more of because we get them from a different lab, I always try and sequence on a flow cell that has at least 1,200 active pores. You can sequence with less than that depending on how many samples you run. However, I think I mentioned it in email, the more samples that you run on the flow cell, the better, because the health of the flow cell is dictated by how much DNA that you have in there, because the pores like to be sequencing at any given point. And so if you load a library and you have like a low amount of library that's loaded in, and you can see based off of how many pores are actively sequencing once you start, that will dictate how long it'll take until the flow cell starts to degrade. And so to optimize that, I should have mentioned it earlier, but I always try and run a full set of 24 samples whenever I can. However, if you sequence at least 12 samples, you should be good to go. But if you plan on reusing the flow cell, I would say aiming for more samples would help preserve the flow cell a little bit more. And usually I run it until the flow cell is dead, and that usually takes 36 hours or so. But I would say if you take some time points along the way, like say 12 hours, 16 hours after you start your sequencing, you can do some quick bioinformatics to see what your general coverage is like, and then determine when you want to shut off your sequencer. But the instructional video in terms of actually using the flow cell, I find that the video that they have on the Nanopore site is pretty spot on to what I usually do in lab. And so I usually just follow the instructional video for that. For the
Sorry, Ralph, I missed the one you mentioned about the 36 hours. Like after 36 hours, the flow cell becomes dead.
Yeah, so what you'll see is that you'll either start running out of ATP within your sequencer, or you start running out of DNA. And so what happens is that if a pore is not actively sequencing something, it will start to decay at a very rapid rate. And so usually around the 36-hour mark is usually when either I run out of library or I run out of ATP in my sequencer. And so you can either add more library or you can add more ATP. But at that point, I find that it's useless because I'll usually get all of the data that I need beforehand. And you'll see that in the graphs that are generated by Minnow over time. And that you'll see that the sequencer will be very, very steadily sequencing at like 80% capacity or more, depending on how healthy your flow cell is. But after the 36-hour mark or so, you'll see that it crashes pretty fast in the matter of a couple of hours. You might go from 80% sequencing down to 10%. And it's very clear when that happens.
So after the 36-hour mark, does that mean we cannot reuse the flow cells anymore?
Yes.
So if we want to reuse it, we have to lessen the sequencing time.
Yes. And so the health of the flow cell can be directly correlated to the amount of sequencing activity that you get in the sequencer. And so you'll see this by the number of pores that are ready for sequencing, essentially anything that's green. So you'll see like a darker green and then you'll see a lighter green in the Minnow. What you'll want to look at is a darker green because that will represent the total amount of pores that are alive and available for sequencing. And so ideally, if you want to reuse your flow cell, what you'll do is you'll stop your sequencing run before it starts to crash. So before that inflection point is when you want to stop your sequencer. And that will ensure that you still have a lot of pores that are still alive and active for sequencing. And then when you do your flow cell cleanup step and then load a new library, because it's getting fresh ATP and everything, it should extend the lifespan of your sequencer for a longer time. And like say if you start to see that degradation, and then let's say that it starts happening very early in the morning, and then when you come into lab at 9 o'clock in the morning, you'll see that the number of active pores went from 80 to say 30%, but it's still decreasing. You might be able to stop it there and preserve them. However, I haven't had a case where, like, if you do the flush, you might recover some flow cell pores from being dead to being active again. But usually I don't see a massive increase even after doing that flow cell wash. And so you always want to stop it before it starts to degrade like that.
So in terms of time frame, like, can you suggest the minimum number of hours that we have to run the sequencing to ensure we get full genome coverage?
Matt might be able to answer that.
Sorry?
Matt,
So are you familiar? Are you familiar with Rampart?
Okay, I'll answer that. Yes. Yeah. So Matt, right now I'm reading up through the bioinformatics pipeline. So, from what I understand, there are like two main protocols. So there's the Rampart and the field bioinformatics, and also maybe. So are we done with the wet lab protocol already? Can we move on to the bioinformatics pipeline discussion?
Yeah, pretty much. So
all right.
With everything else, I follow it as written for the rest of the wet lab protocol for ARTIC.
Thank you so much,
cool.
Ralph.
Yeah, of course.
Really appreciate these inputs.
Yeah, if you have any other questions on the wet lab, just email me, and I'm pretty active on email, so I should be able to respond pretty quickly. So
cool. Thanks.
Let me share the protocol so that you have something to discuss.
Did you want me to share, or?
Oh yeah, if you could share, that would be better.
I can pull it up online. I don't.
Let me make you a co-host.
I'm on Windows partition right now, so I don't have.
Maybe before starting I think the bioinformatics workflow starts at step 16 from the protocol that was shared earlier. So that is when you start the sequencing run using MinKnow. So my question is this MinKnow already provided with the MinION device? Khalil, yes.
You download it
You still download it. Okay.
onto your, yes, no. So let me share. If you go to, this works.
If I'm not mistaken, MinKnow is downloaded from the Nanopore community, which
Correct. So if you
I'm
go
already
to
in.
community.nanopore.com and you go to your
Yes.
dashboard, all your software downloads will be over here in this area. And what we use in our lab is Guppy for base calling and demultiplexing, and
Okay.
the MinION software right here. So MinION software is available for Windows, Mac, and Linux. Guppy, it's available for Windows and Mac, but you have to run on Linux realistically.
Okay.
And I can get into that in a little bit. I did want to hear about what sort of tools you guys have available in terms of computers, servers, anything else. Like what sort of resources do you guys have available bioinformatically?
Right now, because I'm planning to run this on a 64 gigabyte RAM local computer workstation. It has, I think, about 1 terabyte of space. So yeah, that one we can use for storing the data initially. Like these runs, how large are the data sets generated usually?
Usually I'd say 400 gigs or less.
400 gigs or less. All right. Okay.
Yeah, they're not.
Maybe we can.
1 terabyte should be able to store a few runs, unless you're getting throughput that we haven't seen before.
Okay.
Nonetheless, it's not enough if you're doing a multiple runs, and it's not something you want to use as long-term storage. We have 12 terabyte SSD storage on the server and 64 terabytes on a NAS.
Okay.
So
because our current infrastructure is that we have actually like 9 local workstations. Yeah. Mostly of them they have 8 gigabyte. We have 164 gig RAM, but maybe in the next runs I can expand the RAM of the other workstations. Server wise, because our servers we remotely access them through internet, so I don't know if that could do the trick, like because you need internet access, and internet access here is not that good to be quite honest. So maybe for this first run we can do it locally, like run the MinION software, then do the base calling with Guppy. Yes. And do you recommend using the MinKnow software, Matthew? Or
yes. So sorry, you need to use the MinKnow software to interface with the MinION sequencer.
All right. Okay. Khalil, can you share the screen for the? Yeah, I think that's where we will start. The first step in the protocol, the bioinformatics one. Yeah. So okay, so from what I understand, so the sequencing run will be facilitated by MinKnow. I think according to the SOP, the MinKnow does the base calling already. If I'm not mistaken.
Base calling is kind of a sticky issue. You can do fast base calling. Actually, let me share again. I've got a presentation that I actually did on this. I can show you the difference between the two base callers. All right, let's see here. So this is an example of, if you look over here at this image, there's two essential models that you can use to base call with. There's the high accuracy model known as HAC, and there is the fast model. Your fast model is, you can run it on a normal CPU with no problem. You can do base calling. If you've got a powerful enough workstation, you might even be able to do live base calling with it on a CPU. But your Q scores are going to be substantially lower than the Q scores that you will get from the high accuracy model. This was with an R10 using Guppy 4.0. But it's a substantial difference in terms of the shift between, you know, and we actually see with the R9.4, which is what I'm assuming you guys are going to be using, closer to 12 as a median or a mean Q score for the reads. So using high accuracy base calling is absolutely important if you want to turn these into assemblies. The problem is that high accuracy base calling takes a week or more on a normal CPU.
Okay.
However, you can do it in a matter of hours if you have an NVIDIA GPU, which is why you need to be running Linux on at least one computer, because high accuracy base calling is only available for Linux. It's not available on Macintosh or Windows.
Because I'm thinking for the first run, maybe we can do the default Mino run, then do the base calling with the same software, and maybe do fast mode. If I'm not mistaken, so for example, you did the base calling, so you can do both fast mode and high accuracy mode base calling with the same data. Is that correct?
That's correct. So you just need to make sure that you save the FAST5 files. The FAST5 files are essentially they're HDF5 files that are with a special format, and they retain all of the electrical signals, all of the, you can see this is essentially what they store over here, along with a bunch of metadata about the reads, like translocation speed and everything else. So once you have those, you can rebase call at any time. So you can do live fast base calling and then take your FAST5 files and rebase call it with a high accuracy. You can use research base callers like Benito. There's a lot of different options. If new models come out in a year, you can rebase call and get even better results. But the problem is that if you want good data now, if you want to generate good assemblies now, you need to have an NVIDIA GPU because the base calling is it's too slow on a CPU. It's, like I said, it's a matter of weeks versus hours between CPU and GPU base calling.
Okay, I think the 64 gig RAM has a NVIDIA. I'm not sure if it's GPU, but I think it has NVIDIA. Yeah, NVIDIA.
Yeah.
Well, but we'll see about that.
So
okay.
Yeah, I would not recommend anything less than a 1070 if you have that GPU. You can actually do this on a laptop if it's got a decent, like if one of you guys has a gaming laptop at home, you could install another hard drive and use that to base call. I did that a lot with my laptop when I went to Ukraine.
Okay. So the actual sequencing run, so the wet lab people will pipette in the sample onto the Minayon device that is plugged into the computer, and the Minayon device will of course real time do sequencing. So that output would be a FAST5 file.
Yes.
Then we have an option to do like the live base calling, right? If the
I think
system
we should
is...
forgo the live base calling for now because
because we're still testing. Yeah.
Yeah, and you can always go back and rebase call that data later. Just be aware that if you're planning on using these for assemblies and phylogenetic analysis, it's going to be expected that you use the high accuracy model. And that's worst case scenario. We can figure out a way for you. It might be faster for you guys to even transfer the data to us, and then we base call and return you the base called reads. we do have that compute capacity available. It's just about how fast it is to move things over the internet.
Okay. Okay, the multiplexing, I think. So like lately I've been reading about the different bioinformatics pipelines. So this one, I think is this based on the field bioinformatics pipeline? Are you
No,
familiar? Matthew?
this presentation was what I did for the American Society of Microbiology when we presented on the ARTIC assembly process. So there's a little bit of Rampart in here, but it's not the focus of this. Most of this presentation is how the actual assembly occurs, because it's quite a bit different than your standard De Bruijn graph assembler like Fly.
Yeah, because I think this is based on PCR tiling. So I think the assembly would be different. It's not like the normal or the very common NGS, maybe from Illumina or.
Correct. It's a reference based assembly. So you're assembling to a reference genome instead of just, like I said, building a graph out of your reads and looking for overlaps.
All right. Maybe just a few more questions, like on the actual sequence, the bioinformatics run after the sequencing run. So my question is, sorry, Khalil, can you flash the protocol again? Yeah, maybe we can just do that for the reference.
Second.
And then just to re-emphasize for the FAST5 files, because you guys are doing the fast base calling as a first pass, whoever is doing the wet lab for actually setting up the sequencing, I don't know if they've changed it so that it by default saves the FAST5 files, but whoever is setting it up on the computer, you have to make sure that you specify to save that FAST5.
But you don't need to save. There's like an option to like bulk save. You don't need to click that. The bulk save has a lot of other data for troubleshooting that you're honestly never going to need. You just need to make sure you get your FAST5 files and your FASTQ files if you're live base calling.
You guys, I think we are about to overtime. You guys, Matthew and Ralf, are you okay to continue?
Yes.
Okay. So what happens is that so you generate the FAST5 files when you're doing the sequencing. Then the Minnow or the software that's running the sequencing, does it convert the FAST5 to FASTQ right away, or?
Yeah, so if you have live base calling enabled, it will, but just be aware that depending on how powerful your workstations are, it may take you a couple days after the sequencing run has completed for the live base calling to finish.
Okay, yeah, so the FAST5, so it's during the live base calling where the FAST5 gets converted to FASTQ. Is that?
Yes, but the FAST5s are retained. So base calling is the process of converting the electrical signals of the sequencer into A's, T's, C's, G's. So that's
okay. So that's where the FASTQ gets FAST5 gets converted to FASTQ. All right. Okay, understood. Okay, so maybe another question. I think my last one or two questions. So Matthew, would you recommend me doing the Rampart? I think Rampart is more real time, so I think it will involve live base calling. Is that correct?
You can run Rampart anytime, but it obviously works best if you're so you can do it. And so the problems with Rampart are that it's like you can't work with raw data. It can only work with fast queues. So if your live base calling is lagging behind your sequencing speed, it's going to be lagging behind as well. We don't, generally speaking, use Rampart. We got it running to test it, but we haven't actually needed to employ it yet, because the bottom line is you're going to get what you get kind of at the end of the run anyways. I would follow Ralf's suggestions for when to terminate the sequencing run, and then you work with the data you have. Rampart's great for kind of visualizing and knowing which samples are good and which are bad, but you'll find that out eventually anyways. So it's not a strictly needed test. It's original intention is going out into the field. So you're somewhere in a tent with a laptop and you want to know if these samples have the virus at all. It's really great for that, but not strictly needed for this process.
I see. So maybe my last question, last question. So the field bioinformatics, I think that's Rampart and field bioinformatics are different, right? So I think the Rampart, sorry. Okay.
Rampart should be the field bioinformatics.
Okay.
You're looking at the ARTIC, the assembly process is the ARTIC MinION protocol for NCOV19 version 3. Okay. I can share the link to that if you'd like. I've got it here. No, actually, you guys are on it.
Okay, so my recommendation for the bioinformatics run for the first sequencing run is that you will do the sequencing, then do the bioinformatics pipeline after the sequencing run itself, not do the live base calling itself. Right?
You can do the live if you're going to stick with doing an initial run with the fast model instead of the high accuracy model. You might as well start base calling right off the bat. If you don't have the NVIDIA GPU to do the high accuracy base calling, just do live base calling and run with the data that you've got. And we can work on possibly figuring out how to get those resources available later on.
Got it. Yeah. That would be it for now. Thank you. Thank you so much, Matthew.
Okay.
All right. Any last questions before or suggestions from Ralf and Matthew before we end the meeting?
No, do you guys have any more? We haven't really stepped into the bioinformatics too heavily. If you guys have any more questions about it, let me know. I said you pretty much have to be running Linux for this process. I don't know is that what you guys are running on your workstations, or are you running Windows?
Yes, our local workstation computers are equipped both with Linux and Windows. It's actually double booted, but if necessary, I can do the Linux. I can boot it to the Linux during the sequencing run. Yeah,
I would definitely recommend getting all the software set up on Linux. That way, as you can see here, the assemblers, all the bioinformatics tools only work in Linux. Your high accuracy base callers only work in Linux with GPUs. So I would definitely recommend going that route. Everything here is pretty straightforward. Note that we generally run the experimental Medaka pipeline, and these assemblies go very quickly as well. So they don't, these are not computationally intensive assemblies. So generally speaking, you don't need to necessarily use a whole bunch of server power for it. The biggest issue is honestly base calling. But yeah, just copy and paste the commands in and change out the arguments if you need to. It can be a little bit finicky getting the ARTIC pipeline to run the first time.
We'll probably not do live base calling because based on our previous discussions with other collaborators, it's kind of tricky as well. So given the issue on our limited reagents, so I think it's better that we just run the sequencer and then process the data afterwards. Instead of doing a live base call, which have other computational requirements. And
that would be good. Yeah, thanks, Mitch.
Okay.
And then
go ahead.
Yeah, one last thing about the wet lab protocol. So what they don't really mention is the amount of time it takes from like getting the sample to actually getting data. For our workflow, we're able to, like if we're working very hard on it, we're able to get it from raw sample to assembled genome in about a week. But that comes with a lot of factors that come into play as well, especially with the bioinformatics, because that might take a while to do. However, from what I've seen and what I've experienced, the wet lab can be done in 2 days very easily.
2 days.
So what I recommend is if you say get the raw RNA samples that you're going to be redoing your diagnostic sequencing from, I would try and at least convert it to cDNA, doing that first strand synthesis the same day. So I don't introduce another freeze thaw cycle for the CT values.
Okay.
after getting CT values. So what I do is I do my diagnostic PCR, and then I just store my RNA on ice for like an hour, hour and a half, however long it takes until I get those CT values. And then once I have the CT value, I go straight into that first strand synthesis. And so usually what I do is for day one, I do my diagnostics, I do my cDNA conversion, and then I will usually do my ARTIC amplification using the IDT primers all in one day. And then once I have the IDT cDNA amplification with a Q5 polymerase set up, I'll usually leave lab for the day. And then the next day is I just do the entire library prep. For some people, especially if they're first starting out, it can be a little bit daunting because I've found that it takes 4 to 5 to 6 hours to do the entire library prep. I'm not familiar with Illumina, so I don't know how long an Illumina prep takes. But
So we
it's
usually
usually
use 1 day for Illumina.
1 day. Okay. So it might be similar to that. So maybe you're just used to doing that amount of work for library preparations. However, if you wanted to say take a break, like if you're working on additional stuff simultaneously, you can stop the protocol at any point after an Ampere cleanup and just store it at 4 degrees overnight for say 2 days. And so there's a lot of stopping points that come into play. The fastest I've done it is 2 days. And so as long as, say, if you do your diagnostics for your RNA, as long as you convert that to cDNA, the rest of the wet lab protocol is pretty forgiving because the cDNA is a lot more stable.
Thank you, Ralph.
Yeah, of course.
A question
Thank
from
you,
Matthew.
Matthew, as well.
Matthew, would it make sense to do some sort of QA, for instance, like we do the bioinformatics here, and then maybe send a FAST5 file just to see whether we did it correctly? Is it something that would make sense?
I don't necessarily think that you need to share the FAST5 files with us to make sure, because you'll know if you can just run a simple mapping program to see if you're actually getting the sequence you intended. One of the things we didn't mention is post-read quality control. So there are some programs like PycoQC and Nanoplot that you can use to sort of get a more in-depth view of how your run went. And I think I can share some of that with you guys here if you want to see what it looks like. But PycoQCs, they're all pretty easy to set up. Oh, give me a second here. I need it. And then it saves it as an HTML file, so you can just email it out. I'll just share this real quick. Generally speaking, the first thing I do after I base call and demultiplex is I run PycoQC, which will tell me all the information I need about the number of reads, the number of bases, the median length, N50 length, the quality, how long the run was, summaries by barcodes. We know how many reads there are. You can see this is an ARTIC v3 run we did where almost all of our amplicons were very, very close to 500 base pairs. We had a few fragments down here, a few up here. Breaks down the Fred quality scores, and as you can see, we're much higher than that graph I had shown you earlier that was generated using NanoComp. And this is using the R 3.6 model, which should be the current one that's still available. You can run the Benito research model if you want to, but it takes a little bit more work. So yeah, you get a whole bunch of information about everything. And one thing we didn't mention that we should is unclassified. There's a problem with, or some people have reported problems with unclassified reads in the with the current release of Guppy. You can try running it with and without detect mid-strand barcodes. Generally speaking, they changed the algorithm that detects barcodes mid-strand, and it resulted in a much higher rate of unclassified reads. So you can make that adjustment when you're doing your high accuracy base calling. But yeah, this is generally just like baseline QC that we do just to see how the run went. And then from there we can actually do the start the assembly process.
Cool. All right. So if nothing else, well,
Did
thank you
that-
so
did
much.
that- No
Yep.
problem. Did that answer your question?
Yes, yes. I mean, just find a way to see whether, well, first if the wet lab went well, and second if the bioinformatics went well.
Yeah. Generate a Pico QC report, and we can take a look at it from there. That would be the first step, I would say. Just because transferring the FAST5 files could take potentially days, depending on how fast our internet
Yes.
connections are. And we'll only do that if we need to base call the reads for you.
All right. Cool.
All right.
Yeah, I think even the FASTQ files are very big, so I don't know if I'll be able to send you the FASTQ files.
Yeah, and
I think it will take gigabytes. I think the FASTQ files.
yeah, they can get pretty big too. Generally speaking, the assemblies are very reasonably sized, of course, because you're condensing all of that down to single FASTA, but nonetheless.
Yeah.
And always feel free to hit me up on email, and we can do this again if we're running into problems.
A quick question, Paul.
Thanks.
Were you able to get the FAST5 example file from Homer?
I'm doing the install currently. I'll do the test run this week because I've already installed the
Cool.
tools.
So you
I'll
already
do
have
the
the file.
test run today.
Cool.
I will download it and do the test run this week, maybe tomorrow.
All right.
Thanks.
So again, thank you so much, Ralph and Matthew, and Paul and Mitch. So good luck for the first ever sequencing.
Thank you guys.
Yes.
Really
Yeah,
appreciate
thank you.
your
Thank
help
you.
for this.
Yeah, thank you, Matthew.
Thank you
Thank
so
you,
much.
Ralph.
All right. Have a
Take
good day.
care.
Bye.
Thanks. Bye.
Bye.